Good morning, everyone. Thank you so much for joining us. My name is Michael Egan, and I'm the board president of the Myasthenia Gravis Association. I would just like to formally welcome you to the 62nd annual meeting of the Myasthenia Gravis Association. We're so excited to have you joining our program this morning. All the attendees that responded and RSVP'd before November 1st should have received their packet in the mail, and it contains all the information about our schedule today and other really interesting information and accomplishments that the board, the organization has taken care of in this past year. If you did not receive, if you actually RSVP'd after November 1st, your packet is probably likely still in the mail and they'll be coming. If you don't receive it by the middle of next week, just let us know at the Myasthenia Gravis office and we can resend that back out to you. We are thrilled today to have over 240 attendees from 9 different countries, including 50 nurses that are tuning in this morning. This truly shows the impact of virtual capabilities. Most of our program and updates, and as well as our financial status, is found in that booklet that I just shared with you. Normally we go through the financials. This year we're going to just let you review that information that's in the packet that was sent out to you. If anyone has any questions about any of the information in the packet, the financials, just please reach out to us at the Myasthenia Gravis Association, and we will get back to you and answer those questions. Our annual meeting allows us the opportunity to engage and interact with those in the industry. Today, we are fortunate to have Dr. Dimachkie of the University of Kansas Medical Center here to help us welcome and moderate questions from our keynote speaker. And our keynote speaker is Dr. James Howard of the University of North Carolina, Chapel Hill. Dr. Howard will speak on the emerging therapeutics of the myasthenia gravis. I would like to just give a special thanks to our sponsors today, argenx, Alexion, Janssen, Immunovant, UCB, Catalyst, ARJ Infusion Services, and Distinctive Meetings for their generous support in today's meeting. We could not put on this meeting with the help of these sponsors, and actually these sponsors are so important and valuable to us throughout the entire year. So just a heartfelt thanks from me and the entire board for everything that all of the sponsors do for our clients with myasthenia gravis. As we close out the meeting today, we're going to have recognition for some people in our organization. We're going to give out the Volunteer of the Year Award, the Stackhouse Award, which goes to someone who exemplifies the spirit of volunteerism and leadership embodied in the philosophy of the Myasthenia Gravis Association, as well as a special announcement at the end of the day today for the Diana Wilmoth Sunflower Award, which we are awarding this year to somebody who has made a lifelong impact to our organization. Earlier this week, we had a board meeting and we took care of some various things, approving the budget, approving the members of the board. In your packet, again, on page 24, you can find this information. But I would just like to make you aware of the board members for 2023. Those elected to the board as the officers for this year include myself, Michael Egan. Brody Beck will be vice president, Stephanie Hubers, secretary, and Derek Haverkamp will be the treasurer. The additional board members that were elected for 2023 are LaDonna Diller, Lisa Sokovich, Joe Brandt, Dr. Mamatha Pasnor, Dr. Graham Naz, Al Dimmitt, Tom Van Sagi, Jacqueline Carroll, and Lindsay Fleece. These dedicated volunteers make it possible for the Myasthenia Gravis Association to accomplish its mission and to serve over 2,000 people every year. At this time, I'm going to turn the podium over to Allison Foss, the Executive Director of the Myasthenia Gravis Association, and let her give you an overview of what's going to take place throughout today.
Thank you so much, Michael. Good morning, everybody. We are thrilled that you're here and connected with us for our 62nd annual meeting. I have to say, when we decided to keep our meeting virtual this year, I was a little concerned about the people that we serve locally being able to attend. And then when we saw the outreach and outpouring of people from coast to coast and countries to country, it was really cool to see the impact that we're making. So thank you for being here and joining us from the comfort of your home. I first want to say thank you to our support group coordinators. They are located on page 32 of your program. They have continued to roll with the punches over the last year with any of our changes and navigating programming with COVID-19. We are extremely thankful for your volunteerism and your support. Also, just want to piggyback with Michael's thank you to our sponsors, argenx, Alexion, Janssen, UCB, ARJ Infusion Services, Immunovant, Catalyst, and our friends at Distinctive Meeting Group. Again, today wouldn't have happened without all of your support and making this possible. Also, thank you to our staff, Meredith O'Connor, our St. Louis Program Coordinator, and Catherine Singleton, our Patient Care Specialist. They have done a lot of work behind the scenes to help today happen and make everything run smoothly within our organization as we endured a lot of transition over the last year. They're both the friendly faces that you likely see in clinics, and we're so grateful for their work. Thank you. Also want to say thank you to all of our board members and our medical advisory board. Many medical advisory board members are tuning in with us today, and we're so appreciative for the feedback and support that you give us, as well as our extremely engaged board. Thank you so much. Lastly, I just want to say thank you to ARJ Infusion Services. They are assisting us with the nursing CEUs. We have over 50 nurses getting continuing education today, and that is amazing. So thank you. We hope that you'll take time to read through your program and see what we accomplished in 2021. If you recall, our annual meeting looks back on the year prior and then what's to come in 2023. A few housekeeping items we just want to clear up as we begin programming. If you'll utilize the chat feature on your Zoom for any questions that you might have today during the keynote, that will assist us to get the questions to Dr. Dimachkie and Dr. Howard. Following today's presentation, you'll get a survey, and we would love your feedback on today's meeting. Thank you in advance for completing that. Also, I want to say thank you for our patient and family partners who have continued to be financial supporters of the organization. In the coming weeks, you'll be receiving 2023 membership donation information in the mail, and we hope that you'll consider the Myasthenia Gravis Association in your charitable contributions. Thank you for being part of our journey for over 60 years. We hope you'll find today's programming resourceful, educational, and inspirational. We are first going to hear from Lauren Jarman at argenx as she shares a message from them.
Hello, everyone. Good morning, good afternoon. For those of you across seas, I am Lauren Jarman with our argenx Global Patient Advocacy Team. As an MG patient myself, I know how important these conferences are to continue to educate yourself and your community. I'm truly honored to partner with the MGA this morning. At argenx, we have patients engaged at every step of the drug development, and we've continued to work with MG patients as we develop resources for VYVGART. As you know, in December of 2021, the FDA approved VYVGART for adults with anti-acetylcholine antibody-positive generalized myasthenia gravis. And it has been a wonderful year talking and hearing from so many of you and so many stories. If you'd like to learn more about VYVGART, the MGA and Dr. Gordon will be hosting a virtual presentation on December 13th at 5:30 p.m. Congratulations to Allison and her team for such a successful conference. Patients from around the world are benefiting from the work that MGA is doing. Have a wonderful conference and know that the argenx team is working hard every day to advance treatment options for MG patients and other rare autoimmune conditions. Thank you so much for allowing me to participate. Back to you, Allison.
Thank you so much, Lauren. We appreciated your update, and the work that argenx is doing in the myasthenia gravis space is so exciting. We are now going to hear from our keynote speaker. First, I'm going to invite Dr. Mazen Dimachkie to introduce Dr. James Howard. Many of you will recognize Dr. Dimachkie's smiling face. He is one of the partners at the University of Kansas Medical Center in our clinic partnership. Dr. Dimachkie is a professor of neurology. He is the director of the neuromuscular division at the University of Kansas, and he is also the executive vice chairman of neurology and the Department of Research Programming. Dr. Dimachkie.
Thank you, Allison, for this kind introduction. Can you hear me okay?
Yes.
Wonderful. I've been called Marvin, I've been called Mazen, so don't worry about it, you know. But it's my distinct pleasure today to introduce to you Dr. James Howard Jr. Dr. Howard's education started off at the University of Vermont in Burlington with his bachelor's degree that he acquired in 1970 with special honors, following which he went to the medical school in Vermont and went being an intern in Albany, following which he did residency at the UVA, University of Virginia in Charlottesville, in neurology, and was a research associate for a year after that, and an instructor for another year at the University of Virginia School of Medicine, following which he joined University of North Carolina in Chapel Hill as assistant professor, and since then, the rest is history. He had a meteoric rise to the rank of professor of neurology, medicine, and allied health at the University of North Carolina Chapel Hill School of Medicine, where he directed one of the most prolific neuromuscular divisions in the world. And he's also, as part of his research interest in animal studies, adjunct professor of clinical sciences in neurology at the North Carolina State University College of Veterinary Medicine. His research impact is felt throughout the neuromuscular field, and he's in particular a thought leader in research and clinical trials in myasthenia gravis. He has numerous funded active grants. He has more than 180 publications in peer-reviewed journals. He served on numerous editorial board memberships, in addition to being an ad hoc reviewer for many journals. His service is immense, but I'll focus it on service to the myasthenia gravis community as part of his service to the Myasthenia Gravis Foundation of America, being the secretary of the foundation in 1991 to 2001, the vice chair of the board between 2001 and 2003, and the chair 2003 to 2005. He currently directs the Myasthenia Gravis Clinical Trials Unit and Translational Science Research at University of North Carolina. He's by far the most impactful MG clinical trialist that has advanced the field of MG therapeutics. He's not only a superb researcher, he's a clinician educator that has trained and graduated a large cadre of neuromuscular and EMG fellows. And of course, he's a sought-after lecturer. Why else would he invite him today for this presentation? But this is for national and international meetings that he's sought after. Without further ado, and I could probably spend another half an hour telling you about Dr. Howard's accomplishment, but rather than do that, without further ado, I present to you my friend, my colleague, those that know him closely refer to him as Dr. Chip Howard. Chip, welcome.
Thank you, Mazen. Good day to everybody. And I appreciate the opportunity to speak to you today. I guess my political pact in funding Mazen gave me a great introduction and welcome. So thank you again. We're going to focus today on emerging therapeutics. This all started around 2007, and it has been a meteoric, and that's the term he used, a meteoric rise in what we've been able to do in the emerging therapeutics for myasthenia. And I want to share with you that odyssey, some of which you may have heard before, others of which it will be fairly new. And for some reason, I can't advance. Oh, there we go. So here are my requisite disclosures. And as you can see, I receive research support from multiple sources, including the pharmaceutical industry, and I consult with many members of the pharmaceutical industry, primarily in the development of their phase 2 and phase 3 clinical trials for myasthenia. I'm going to keep this very generic, but we'll be pointing out selected compounds. At this moment in time, there are only 3 approved drugs to treat myasthenia. Imagine that we have gone since for centuries with no FDA-approved drug until October of 2017, when eculizumab came on board, followed by efgartigimod in December 2021, and most recently ravulizumab in April of this year. You will hear other names: batoclimab, Descartes-8, analizumab, nipocalimab, Rosie, as we call it, and zilucoplan. These are all investigational drugs under study and not available for therapeutic use by the general public. And then other drugs that I'm going to mention are FDA approved, but we've been using them in an off-label fashion to treat this disease. And we've simply borrowed what the transplant world uses and have moved it into our domain for treatment. Some of these comments may well be familiar to you. I think it highlights why we need to do more and to do better. I'm in the ER. All my tests are fine. I have shortness of breath, cough, chest tightness, fatigue. I'm scared, upset. I don't feel good. Just because someone carries it well doesn't mean it isn't heavy. And then there's some others that are very telling. I'm what a person with an invisible disease looks like. How many times have you heard that? AChR, antibody positive generalized disease, is having to take a break between putting out different garbage bins because carrying the green boxes plus walking to the end of the driveway is too much exercise for me. Very common comments that we hear in clinic and tell us that we're not doing better. This was brought to the forefront last October when Carolina Barnett at the University of Toronto wrote this editorial. People with MG are better, but are they really doing well? And that's the underlying theme that we're trying to address as we move forward. And so we're going to talk about not only burdens of the disease, what does the weakness do to you, but burdens of treatment, which I think is to a larger degree more important. And we'll speak about these. We clearly have done well. Back in the early 1900s, nearly a 100% mortality. And when we had assisted ventilation and antibiotics, a precipitous fall in mortality to less than 50%. And then beginning in the mid-60s with the advent of ACTH and the corticosteroids, we had further decline as we improved strength. And then we've whittled away, chipped away at this with bringing on new immune suppressive therapeutics, methotrexate, azathioprine, cyclophosphamide, the cyclophilins, and then more recently, mycophenolate mofetil in our new targeted therapies. So there's no question that we've done well. People would argue that more than 25% of these have unmet needs. We know that 8, 10, 12% of people are refractory to our current therapies using our current toolbox, but more than that have certain unmet needs. And I'm going to argue that that number is up around 60%, 70%. Our current toolbox looks like this. And we have symptomatic therapy with cholinesterase inhibitors. I'm not convinced that's a real treatment. It helps the disease without question. Some muscle groups, other muscle groups, it doesn't help. Other muscle groups, they're actually made worse with each dose, and we have to titrate. And this can be quite difficult if one is infected or during menses, pregnancy, things of this sort. We have a broad range of immune suppressive drugs here in the US. Many of these are not available in other parts of the world due to the regulations of their health ministries. And so they don't have the plethora of tools that we have. And we have surgical thymectomy, which is important because we think this is where the break in tolerance to our immune system occurs. And then we have some newer therapies that are coming on that we'll speak about. This is a data cut through the MGFA's patient registry, looking at, on the left here, the myasthenia gravis activities of daily living score, and the larger the number, the worse the symptoms are for the individual. And one can argue the reasonableness of using registry data, and this is a single point in time, but half of the individuals, more than half of the individuals, complained of scores that were above 6. And this is moderate to severe symptoms. So it tells us that we're not doing well. They're not doing well. We as clinicians haven't been doing well. And then when we look at a quality of life instrument here on the right, specific for MG, developed by the late Ted Burns at the University of Virginia, one sees this broad range of impact on their psychosocial well-being, and larger numbers are worse quality of life. And so both of these insights into patient responses tells us that we have an issue. We're well aware of the treatment burden as clinicians, and many of you as patients, in terms of the side effect profiles. And I'm not going to go into each and every one of them, but we look at problems that occur acutely or that are inconvenient, and then those that carry systemic toxicity risk with steroids, mood disorders, the risk of infection, weight gain, osteoporosis, diabetes, and then with some of our other immune suppressive drugs, impacts on our kidneys, on our liver, on our bone marrow. And plasma exchange and IVIG are not without their potential risk as well, with problems with renal function or clots, strokes, heart attacks, etc. And one can see this large list that tells us that the adverse event profiles that patients are exposed to are not without consequence. What else does MG do? Well, clearly it has an impact on daily life, frequent hospitalizations, frequent outpatient visits, depending upon how one is doing. The effects on professional life and income, and I'll show you a little bit more of that. Depression, anxiety, decline in social functioning because they feel embarrassed. They feel that they don't want to put themselves into a situation where they become unstable, and I use that term broadly because of acute weakness that might develop. If they're out enjoying themselves for an evening and then suddenly develop weakness, they may not or are not close to home and don't have that particular support system. So a very large burden of disease that has wide-ranging physical, social, psychological impacts on them. And then we know that, we've just spoken about some of this, that increased feelings of anxiety and isolation because of their concerns. We've talked about the long-term treatment risk. We know that patients with MG can have myasthenic crisis, the need to be on an assisted ventilator. That can be very dangerous to the individual, and it can be precipitated by a number of reasons. Most commonly in my institution, it's because they've been given an inappropriate drug, usually an antibiotic that we know is toxic to nerve muscle communication. And these things are a major concern to folks. This is data that has been developed over the last year by colleagues, and it looks at the rates of unemployment and lost income around the world. In the US, we cannot get good information because of the way we gather data. We're too democratic, if you will, and it's all fractionated. But in Germany, 28% of patients with MG retire early. Japan, 27%. Australia, almost 60%. Thailand, almost 60%. And then many others have a reduction in income because their job has been changed because of their weakness, or the number of hours they're able to work have been reduced. And this has a tremendous impact on the ability to provide for themselves, to provide for their family, etc. Very sobering. And it tells us again that, yes, we've helped strength to a large degree, but there are many elements of the total picture that need to be addressed. And these are the reasons why there's this new strong push to develop newer therapies with much more restricted adverse event profiles that are easily administered and work rapidly, etc. When we look at dissatisfaction, the biggest complaint of patients are side effects to the drug. More than a third if we look at any therapy. And if we look at steroids, that number is in excess of 80%. And this is what we do to people. People are not happy with the fact that when they start a drug, it takes months before it even starts to work. It may be more than 18 months to 3 years with one drug for it to reach full effect. And if one has to cycle because they've not responded to a drug and they've been given a fair shake, one sees the accumulating time lost, as I call it, for life. And so there's this call to arms. And then we have the logistics. People have to go somewhere for apheresis, plasma exchange, or for IVIG, or somebody, as one patient put it, they invade my home to give me my IVIG, and I don't like people coming into my house that I don't know well. And so these are other concerns that folks have. To understand where we are going, we need to know what are the mechanisms that are causing MG. And I'd like to review that briefly with you. Unlike most and many autoimmune disorders that have one mechanism which causes the problem, MG, again, a snowflake disease, has three, at least, maybe even more. But the three most common ones are here. One is that we block the receptor sites for the neurotransmitter to interact. So antibody binding to the acetylcholine receptor blocks the ability of neurotransmitter to get to where it has to go. And there are two places where it has to go. And both of those places have to be occupied by acetylcholine, neurotransmitter, for that ionic channel to open and allow that muscle fiber to have the potential to contract. The second is that as antibodies bind, they sort of cross-link with each other. And that accelerates a normal turnover process. Our receptors on the surface of muscle periodically are internalized, they're degraded, and new ones get popped back up. It's somewhere between 7 to 10 days, depends on the specific muscle, depends upon the species, whether a human, a dog, cat, a mouse, a cow, whatever. So there are these differences, but this gets turned on to work more quickly. And the net result is that we lose the total number available, and that increases the probability that we're not going to be able to energize that muscle fiber so it contracts. But neither of these muscles explain what I'm going to show you a bit later. But we need to talk about how can we manipulate this system for betterment. And one is based on observations that are not new. They're actually old. Sir Francis Bramble, back in the late 40s, identified that there was a receptor process called FcRn, and that term wasn't there when he described it, it came on later, but he found that in the placenta there was a mechanism to transfer maternal mom's IgG immunoglobulin to the fetus in the last 2 months of life. And this is how infants have an immune system at birth, that they get mom's IgG. And then over several months, sometimes longer, that wanes and the infant builds their own immune system. Sally Ward, an immunologist who was working at the time at Texas A&M, she's now in Southampton, England, identified this FC receptor, as it's called, FcRn, that it wasn't restricted to the placenta. It was in multiple different kinds of cells. And she identified it as a salvage pathway for a means of preserving IgG, leading to a very long half-life. And she began working on a group of compounds called ABDEGs, which means antibodies that enhance IgG degradation. And then this was picked up by two folks, and the story behind it is actually quite amusing. We don't have time to go into it. But Hans de Hart, who was the chief scientific officer, and Peter Ulrich, who's the principal scientist, developed the first antagonist to FcRn. It's a fragment of IgG1 that binds very tightly and is now in use, as you know, as efgartigimod or Vyvgart. Old technology, a sudden serendipitous observation leading to the development of a new drug that we find is going to be quite beneficial to our patient population. And so how does it work? This is a cell here on the right. And look at the left panel of it. And IgG, these little Y-shaped molecules, are taken into the cell, and we call this an endosome. And then they bind very tightly to FcRn. These are little orange things. Because they're bound, they get trafficked back up to the circulation, to the cell surface, and re-released into circulation. And this goes on over and over and over, and that's why these things live so long. Those that are not bound, as shown here, can't get trafficked back, and they end up in what we call a lysosome, which destroys them. So if one can block these using an FcRn inhibitor, such as efgartigimod and others that are in development, the majority of the IgG cannot be taken back to the circulation, but ends up and gets degraded here. And this is a 100,000-foot view, and there's a lot more detail, but in a nutshell, this is how this compound is working. As a result of these observations by Professor Ward and then with Peter Ulrich, et cetera, a phase 2 trial was performed that showed a signal, and we designed what was called the ADAPT study, looking at the first-in-class FcRn inhibitor, efgartigimod, as a therapeutic in MG. The study was very unique in two ways. We looked at patients who had mild, moderate, severe generalized myasthenia. They were AChR antibody positive, but we convinced the FDA to allow a small portion of individuals to be AChR antibody negative. These included people who had MuSK, who had LRP4, or had no detectable antibody by the assays that we use. And they had to be on stable therapy, and they had to meet criteria using this activities of daily living score. And they were randomized. We split them. The computer did this 1-to-1 to receive either drug or a matched placebo. And then they were given 4 weekly infusions followed by an observation period. So this were cycles. This is the second unique feature of this trial. In overall, all trials, one is dosed continuously based on focus groups with patients that, like Lauren has described. The message was, we want independence. We don't want to be treated if we don't have to be treated. So he said, okay, this can serve 2 purposes. Let's meet the requests of the patient, but also let's see how durable this kind of a treatment is. If patients finished the trial, they went into an open label extension, and 151 of them did so. And this study has just wrapped up. People could be retreated if, one, they didn't respond to therapy over 8 weeks, or if their ADL score, after improving, crept back upwards to within 2 points of what it was before they started to get the drug, and then we could retreat them. And let's see what happened. Patients were evenly matched when we look at all the various characteristics, other than those who had severe myasthenia, what we call class 4. And you can see that they're underrepresented. And this is true of all trials. These folks tend to be brittle. They tend to have more frequent exacerbations. And from a research perspective, a clinical trial perspective, are potentially dangerous to the validity of the trial because they would increase failure rates. And so when we recruit, though, we try to, those tend not to meet our criteria. So it's heavily favoring these other 2 disease classes. So we treat them at week 0, 1, 2, and 3, and look what happens to antibody and IgG levels. IgG in green, antibody in blue, very rapid fall. That reaches a nadir about 1 week after their last infusion, and then slowly comes back to baseline. And these 2 top curves are the placebo arm. They're not getting active drug. And we're able to show that more than 60% of the IgG is cleared, nearly 60% of the antibody is cleared. And regardless of the subtypes of IgG, what we call subclasses, all of them are affected in fall. We do know that patients who were AChR antibody negative, their IgG levels and what antibodies they had for those that had MuSK, et cetera, followed a very similar pattern to what we see here. What did not happen is that we did not affect albumin. Some of the drugs do, and we did not affect cholesterol, which some of the drugs do. And so this was a very pure IgG antibody response. And how did we do? Nearly 70% met our primary endpoint, which was called an MDADL responder. And what's that? It's an individual who had a 2-point change, 4 successive weeks of that change that had to occur within a week of their last infusion. And so rather than looking at what the ADL score was at the start and at the end, we made it more stringent and said, can you sustain this response for a month? And nearly 70% did so. Here's the placebo arm. And we see these kinds of placebos in all of our MG trials, and there are a number of reasons for it. Another outcome measure we use is the quantified MG score. And I'm sure those of you who participated in trials have undergone this. Some clinics use this intermittently, and they too showed a very favorable response. And what was also very exciting was that of those who responded, nearly 85% did so in 2 weeks. Not months like our other therapies, within 2 weeks. Fast, rapid response. And so this was faster than I see with IVIG. It's faster than what I see with plasma exchange. We're able to see responses even after 1 infusion. Is it repeatable? Here's what I just showed you on the left. Here's if they got a second cycle, virtually identical response, both in the active drug arm and the placebo arm. And so it is reproducible, and graphically it's displayed on the right. And some may argue that perhaps in cycle 2, we have a further reduction in their ADL scores, additional benefit. And this may be true. This is still being determined and analyzed, if you will. When we look at the open label data, we see that the responses are pretty repeatable. And here's the ADL score, here's this quantified MG score, and over 5 different courses of therapy, on average, these patients had very similar responses, suggesting that patients will continue to respond. It doesn't wear off over this observation period, which was running about 3 years in time. So how long does it last? Well, more than half had responses that lasted in excess of 8 weeks, more than a third in excess of 3 months. My longest lasted 46 weeks before she had to be retreated. None of our other therapies that are intermittent, IVIG, plasma exchange, etc., do this. Yes, 10% had a short response, and another third had an intermediate response. But we're now seeing the potential for precision treatment. Can we tailor a treatment to a specific individual and accommodate their wants and their needs? When we look at the safety, much of it is nasopharyngitis and headache and nausea, diarrhea, and this is the placebo arm. Here is the treatment arm in this first blinded phase. And in many instances, the placebo response or adverse events are more frequent than the treated arm. There is concern that is there an increased risk of infection? And this is in the product label as mandated by our regulatory agency, the FDA. But when we look at the open label, the long-term open label, look what happens to these incident rates. They fall drastically. And so we don't have the full picture yet. My personal belief is that it is not going to be a significant issue and that infection risks will be very minimal and not be a major concern for our patients, but very well tolerated. These adverse event profiles are better than any of our prednisones, our mycophenylates, our azathioprins, cyclosporins, tacrolimus, etc. We also have a sub-Q formulation of the same drug. And so efgartigimod, Vyvgart, is administered intravenously. We now have underway a subcutaneous formulation. It's 5.6 cc's, so it's still given by injection, not by an infusion. And we combine it with another compound that breaks down some of the collagen barriers underneath the skin to allow this volume to diffuse. And this has been used in the oncology space with their therapies for several years. It's very well tolerated. And so the question became, is it as good, worse, or better than the IV formulation? And one sees, here's the sub-Q in green, here's the IV response in blue, and it's virtually identical. And here it is graphically, looking at our antibody levels and our ADL scores. And so it's very comparable. And we look at other outcome measures as well. And so the two are comparable. And this will go to the FDA for regulatory approval sometime after the first of the year. So stay tuned. But what it's going to give is choice. It's going to give freedom. So people don't have to be wedded to an infusion center or someone coming into their home. They can take the drug with them, go on vacation, go on business, take their dose when necessary, and allow this improved quality of life. There's been a lot of discussion of why this was not approved for the antibody, AChR antibody negative patient. And so what we found was that those patients who are AChR negative, when superimposed on those who are AChR positive, their response was virtually identical. But we had to compare AChR antibody negative patients to those on placebo who are AChR negative. And that placebo group had much better response, if you will, than those who had the antibody. So the two placebo groups did not patch. The two drug groups did match. And because of this, the FDA would not approve it for use in seronegative. I will tell you in Japan, it is approved and it's being used, and investigators, as well as the company, are gathering data and responses in order that we can seek additional approval, but I have no timeline for you. So we believe it's efficacious. We just now have to prove it to the regulatory authorities. There are other FcRns. The next one that will be most likely approved is rosanoliximab. In the My Caring study, one sees very similar responses to IgG levels and antibody levels. These were 2 different doses. I don't have all of the data for this trial because I was not a part of it. I can only tell you what's been presented at national meetings, but the reduction are similar. The safety and tolerability may be a little bit different, and headaches may be an issue, though we don't know for sure, and we're still waiting to see what the published literature will say. But it too seemingly is effective. Now, this is given subcutaneously periodically, and so it's a different formulation than the ADAPT trial that I showed you previously. And there are several others in the works. There is a product, nipocalimab, batoclimab, and these are all still in early trials, and we have no data from them as well. Let's switch now to complement. And we talked about the different mechanisms of how the antibody interferes with nerve-muscle communication, but it can't explain what I'm showing you here. On the right is a normal nerve-muscle junction. To the left rather, and to the upper right of that is the nerve terminal. Here's the muscle below that. And one sees these peaks and valleys. This is the neuromuscular junction. And it's built this way by nature in order to pack receptors in high density on the crests of these folds. If this were simply a flat membrane, our ability to do that would be much less. And this dark staining shows where these receptors are. But to the right, you see a very end-stage myasthenic muscle fiber. It has none of these post-junctional folds, maybe a little semblance here. And so the receptors are essentially gone. And what we now know is that complement is activated by the binding of antibody to the receptor, and then complement goes through a cascade combining with other complement proteins to form this ring-like structure. We call this MAC, membrane attack complex, or terminal complement complex. And this complex attaches to the muscle fiber itself and literally drills holes through the tissue here, leaks its content, and renders it afunctional. And over time, it architecturally destroys this highly organized anatomy into a flat surface, a rolling hill, if you will. And as a result, the muscle fiber cannot be contracted because it doesn't have enough of these receptors to generate the electrical charge. The first drug approved to treat MG was eculizumab. The study was called REGAIN. This all began in 2007 when we approached the company to see if we could try it in MG, and they agreed. The blue is the active drug, the red is the placebo arm, and again, you see those who get sugar water, if you will, do get better in clinical trials, but the active drug clearly worked very quickly within weeks and then sustained itself and did so much better than the placebo arm. This vertical bar represents the rollover from the blind portion to the open label, and one sees the placebo arm has a phenomenal response as well, and then the two parallel each other out over time. This is the same data, but I've sort of redistributed it. And so the blind portion of the study is below this horizontal dashed line, and above is the open label. The bluish purple is active drug, pink is placebo, but let's just focus to the right side. By the end of the blind phase, 61% of patients were better. But over 130 weeks, 85% of patients got better. These were patients that we call refractory. They had failed other therapies, at least two, or at least one, but required plasma exchange and IVIG. And then when we look at those who achieve an even tighter improvement definition, minimal manifestations, which means no functional weakness, 25% increasing to nearly 70%. And trying to explain that, all we can think about at this point in time is that we're rebuilding our neuromuscular junctions. Those that had been destroyed by this membrane attack complex, we can actually recover them, rebuild new ones, and get the muscle fiber working. Now, we have no data to support this. This is an area of active interest that's being looked into. It's simply a hypothesis, but a very intriguing one for us. This is a cousin. This is ravulizumab. This is the third drug that was approved by the FDA, and one sees it's a very similar response. Red is placebo, blue is active drug. The timeline is expanded, so it looks like it's not as impressive, but it is. One sees a rapid drop that's sustained, and here the placebo on rollover also develops an excellent response. And so these two are similar. Ravulizumab is a cousin. It's actually eculizumab. It's been engineered so we can administer this every 8 weeks rather than every 2 weeks. Again, quality of life measure. And then there's another one that the study has been completed. This is going to the FDA at the end of the year. And this is zilucoplan. This is not a full-size antibody. This is what we call a macrocyclic peptide. It's only 14 amino acids. It's administered once a day under the skin, much like we do with insulin on a daily basis. And we looked at its response over 12 weeks, a short timeline given what we saw with Regain. And one sees, again, this very similar response, rapid, robust, and sustained. And I will tell you in the open label portion of the trial, which is still ongoing, the data cut that we've just completed shows that this is continuing on, and the placebo arm comes down and follows it as well. And so given that this is subcutaneously administered, now has the potential, if approved, to offer freedom, travel, convenience to our patient populations. So we're listening to the patient, trying to develop these tailored programs that we can use. I will tell you that we have just completed a pediatric trial with a complement inhibitor, eculizumab. This was just completed a few weeks ago. It is going to the European Medicines Agency at the first of the year, but this is a very familiar response. This was an open label. There was no placebo arm, so I can't show you that. But if you superimpose this to the original Regain trial, it's virtually identical and sustained. And so we now have a drug that is available, hopefully, to our pediatric population. We have pediatric trials that are underway with efgartigimod, Vyvgart, and we are beginning pediatric trials in ravulizumab and zilucoplan in the next few months. I want to also go back to the antibody and tell you a little bit about what else is going on. And we believe the antibody plays a prime role, complement plays a critical role, but there was a US study called BeatMG that looked at a drug we've been using for years called rituximab, and it failed. It failed in part because of study design, and this has been argued, but I think it was compared to the placebo arm. The placebo arm was prednisone, and the ability to reduce prednisone, and it didn't show much of a difference. And I think people need to learn their lesson that you never compare anything to placebo, to prednisone. The mycophenolate trial failed. The methotrexate that Mazen's well aware of failed because prednisone was involved. It's too good of a drug to improve symptoms, despite the fact that it has horrible side effects. There is a recent Swedish study just out in the last 2 weeks that shows that it may be favorable. So if this is truly study design, there still may be a role in AChR-MG. It's clearly our drug of choice at the moment for MuSK-MG, though I think FcRn inhibitors may alter that once we get a little more experience under our belt. And it's possible that rituximab doesn't work because it targets a marker on our lymphocytes called CD20, and they're only expressed on a very small number of the family of what we call B cells. And so there's a trial with a drug called inalizumab. It's already approved for another disease called NMOSD, and it is expressed on a larger number of cells. And we have no data. This trial is still ongoing, but should it work, that may offer an explanation of why rituximab doesn't work as well. The other one I want to just mention to you is targeting what's called B-cell maturation antigen, or BCMA, which is expressed on plasma cells. And it's through a program called CAR-T, chimeric antigen. And what we do is we take cells from patients, we harvest them, we ship them away. They are re-engineered to change their structure, and then we give them back to the patient. Now, you've heard of CAR-T in terms of cancer therapy, and that uses our DNA. And it's pretty tough drug in our program because of the side effect profile. 5% mortality, nearly 25% have an acute severe respiratory illness, 15% or so have neurological complications. This program doesn't use DNA, it uses RNA, which is the pre-structure of DNA. And it doesn't have these kinds of side effects as best we can tell. And so DNA-based is continually, it's proliferative, and this is why we have toxicity. This RNA-based therapy is self-limited. It turns off after a while, and so it doesn't proliferate ad infinitum. Now, it has good points and bad points. We don't have the side effect profile at all, but it may not have lifelong response that we might potentially get using DNA-based therapy. And there's a trial, it's called DESCART-08, that's undergoing. We're looking at different infusion parameters, and we're going to begin a phase 3 trial in the next several months with this compound. And so we've made progress. There's absolutely no question. We now have targeted immune suppression with complement inhibition, with FcRn inhibition, and we're reducing side effects. We're improving the rapidity of the onset of action. 2 critical complaints of dissatisfaction with our patients. We're now able to treat those who are refractory to other forms of therapy. Not all of them, but many of them. And so we can improve their quality of life as well. We now have different formulations, IV, sub-Q, in both classes of therapy, hopefully within the year, that will allow us to give choice to the patients and give freedom for movement around. And we're starting to develop data that looks good in our pediatric population. But we still have a couple of areas that we need to address. One is, what's the safety in pregnancy and childbearing patients? What's the safety in lactation? There is some data for complement with another disease called PNH that's coming out of Britain, and it appears safe, but we have no data in the MG space as yet. Can we use this for people who acutely deteriorate? We currently use plasma exchange or IVIG or increased prednisone, and can we now use things like complement or FcRn inhibition because it works so quickly that we can now start to use them? And given their side effect profiles being very nominal, we are now able to improve upon that as well and get them out of the intensive care unit or prevent them from going to the intensive care unit. So the future looks great. I didn't show you a slide of what's in the pipeline. There are at least 15 other compounds targeting various aspects of the immune system that are coming to trial or just starting early phase 2 trials that we'll have to see how it goes. My plea to you folks is that if you have the opportunity to participate in a clinical trial, consider it. We've just completed a study looking at racial and recruitment diversity in MG trials in North America. And there are huge swaths of the country, particularly in the upper Midwest, in the upper part of the country, and in the South, where we have very little recruitment into clinical trials. And there are a number of reasons, and it's not patients' faults. They don't have access in part to study sites and things of this sort. So it behooves sponsors to transport patients in who want to participate. But just because you live in an area that doesn't have active MG things going on doesn't mean you can't participate. So reach out if you're interested. We're going to need many patients because we can't use the same patient in different trials in 99% of the ones that we have running. So we're seeking yours, and it's your generosity by participation that has moved this bar forward. It's not us, we simply do the mechanics, but it's your willingness to participate, your generosity. And it is a hassle with childcare, et cetera. We recognize that, but we thank you for doing so, and we look forward to many more of you participating. And with that, I'll stop talking and answer questions. Thank you very much for this opportunity.
Dr. Howard, thank you so much. That was a wonderful presentation, chock full of good information. We are going to take a 10-minute break. Please put your questions in the chat. We are monitoring the chat. And when we come back, we'll hear from Alexion, and then Dr. Dimachkie will help moderate the questions with Dr. Howard. So we'll see you back in about 10 minutes. Thank you. Welcome back, everybody. We are going to hear from our partners at Alexion in a message from Kathy Logan.
Hello there. My name is Kathy Logan, and I'm a patient education manager here at Alexion. Thank you all for joining today for the annual meeting. I feel so honored just to have a few minutes of your time to talk about Alexion and OneSource, our patient support program. Every day, those who live with rare diseases and devastating conditions really inspire us to push the boundaries of medicine, technology, and healthcare services. Our goal here at Alexion is to transform their reality for the better. Alexion is headquartered in Boston, Massachusetts, and serves patients in 50 countries. Alexion has 5 approved medicines covering 7 different rare diseases and devastating conditions. There have also been over 40 clinical trials to study complement-mediated conditions. As you may know, OneSource is Alexion's complementary personalized support program, really offered and designed to support your specific patient needs. This program has been transformed to create a seamless and more specialized experience for you, the patient, by incorporating new technologies and really building and scaling to better meet the needs of our patients. As part of OneSource, the patient education manager, such as myself, will provide that patient education and lead different regional and local patient initiatives. We are also able to provide that site of care support and education. The patient navigator will be the point of contact initially within OneSource and also assist patients, helping them to understand their insurance benefits, and also will be there to support the patient really from OneSource enrollment through at least their first infusion. Patient support is typically then transitioned to the patient liaison, who will then provide that ongoing education and support for patients who then may have life event changes or insurance changes down the road. OneSource has 4 different areas of focus. Those include providing disease state education, navigating the health insurance process, connecting patients within the community, and also just providing that ongoing support. OneSource is here to help so that you're not ever alone in your treatment journey and will be with you every step of the way. I want to thank you all again for joining, and feel free to reach out to me with any questions at the number listed on the screen, or you can refer to the OneSource website at alexiononesource.com for more information. Thanks, everyone.
Thank you so much, Kathy. We appreciate having that information and hearing from Alexion. I'm going to turn it back over to Dr. Dimachkie and Dr. Howard Dr. Dimachkie is going to moderate the questions that you all have entered in the chat. There is still time if you want to enter in any questions or if anything comes up as we discuss. Dr. Dimachkie.
Oh, thank you. Thank you for turning over back to me. We have several questions here in the chat box. I'm going to start with the first question here, and I have a follow-up to it. The question goes as follows. As I understand it, none of the new 3 drugs treat MuSK-MG. Am I correct, Dr. Howard?
No. Complement inhibitors will not treat MuSK-MG. MuSK is mediated by what we call an IgG4 antibody, and IgG4 does not activate the complement system. FcRn inhibitors, on the other hand, clear all of the subclasses of IgG, IgG1, 2, 3, and 4. And so there is some small amount of data already, and theoretically, we should be able to treat MuSK-MG with an FcRn inhibitor. And then several of the newer drugs in the section on B-cell depleters that I talked about, inelizumab, for instance, and some of the newer ones that will be coming to trial have potential to treat MG and are currently incorporated into the study protocols that will be forthcoming.
Thank you, Dr. Howard. A follow-up to that question, is there a reported clinical experience or other experience to indicate that perhaps, as you indicated, the FcRn blocker currently on the market might be beneficial in MuSK-MG? Has there been approval hurdles? Sometimes insurance companies can determine who gets what medication. What's your general comment about that?
Yeah, so patients who had MuSK-MG or LRP4-MG were included in the non-AChR positive arm of the trial. And there were 6 MuSK patients in the ADAPT trial, 3 getting drug, 3 in the placebo. So the numbers are exceptionally small. On the face of things, this AChR negative population seemingly did as well as the AChR positive population. And we would assume that they would be responsive. It was the placebo arm that negated accurate determination of the efficacy of the drug. I am not aware of it being used yet in a patient with MuSK, though I'm not privy to all of the prescriptions that have been written for it. And it's possible that someone has convinced their payer to allow it. But theoretically, I would expect the MuSK patient to respond much like the AChR positive patient.
Yes, and with small numbers of patients of 6, half of whom received the drug, half received placebo, you need much larger numbers typically. We need much larger numbers typically to demonstrate an effect, and this would be just a spurious variability in the placebo group that just created this non-separation between the 2 groups. But at the scientific level, the drug we think should work, though again, the proof is not there from research studies. Yeah.
Correct.
For the MuSK-MG subpopulation, yeah. The next question is about combined variable immunodeficiency, CVID, hypogammaglobulinemia, and how does Vyvgart work in that context of CVID hypogammaglobulinemia?
Well, individuals who are hypogammaglobulinic, we would not want to be treating them with Vyvgart because it would be further depleting their immunoglobulins. And I'm no expert in that disorder, but would believe that it would not be efficacious in that group of patients.
Thank you. Switching gears to about rarity of the disease. This is more of a disease state question. MG is considered a rare disease with more awareness and better diagnostic processes. Do you think it will still be classified as a rare disease?
Our best data to date says that it is a rare disease and probably will be a rare disease. But there is no question that education to the physician community, education to the patient community, social media have increased our ability to identify patients with myasthenia. And so if the rare disease is less than 200,000 patients and we were at X level, we're up a bit, but we still haven't exceeded it. So I don't see it becoming a common disorder. I'm not sure I want it to, because that will have impact in terms of governmental assistance programs, other interactions with insurance companies, research, et cetera. So, but we do clearly need to identify everybody in the community who has this disorder, and we're getting better at that.
Thank you. Going back to the theme of CVID, and this is a rather specific question, and typically, I'm not sure your style, Dr. Howard, but typically, if it's a very specific about management of a patient, I try to frame it into more generic terms that will be more generalizable to the global audience. Are you okay with that approach, or do you prefer reading the specifics of the question?
No, that's okay. But again, I'm not sure I can answer that question of that particular disease.
Yes. So this is a question, the triple seronegative, single fiber EMG positive, AChR binding is moderate, it says. General MG in patient with significant breathing issues and weakness plus CVID. Neutropenia associated with cell sept, concern about rituximab, plasma exchange, currently on IVIG, seems maximized, patient still very debilitated. Any suggestion to a theoretical case scenario, not specific to this patient?
So there are individuals who are really tough for us to manage, having failed selected therapies, have comorbidities or other issues that impact on our choice. And so one has to take a very thoughtful approach as to what the options are and the impact of that therapy and response. Clearly, we don't want to be depleting someone of immunoglobulin who's already depleted in immunoglobulin. And so selected therapies are off the table. There are perhaps other comorbidities. For instance, if one had diabetes, I'm not wanting to give them high doses of corticosteroids. Or if they have renal disease, I'm not wanting to give them a cyclophilin, cyclosporin, tacrolimus, because of the potential impact there. Or if they have liver abnormalities, drugs like azathioprine may not be a beneficial. And so I've always preached that myasthenia management is not a cookbook. It's tailored to the individual. It's tailored to their pattern of weakness, the rapidity of which the weakness occurred, their comorbidities, and unfortunately in the US, their financial wherewithal, because they carry a burden of cost, unlike many countries where it's government funded and dealt with. In a situation like this, if they're AChR positive, if I've heard correctly, one might consider a complement inhibitor. It would not have impact on the other comorbidities that I've spoken about and may well be beneficial. And so that would be something to consider.
Thank you, Dr. Howard. There's also another specific question that I'll paraphrase a little bit. Someone who's AChR antibody positive, unfortunately developed skin cancer on the scalp, is immunosuppressed. His cancer or her cancer now has become inoperable. They're concerned about chemotherapy possibly being harmful to them for the treatment of their malignancy. And the question is about the prevalence of cancer of this type in MG patients. And are there any successful treatments? Though, again, we're not cancer specialists, so we're not going to profess towards that,
Yeah, and without knowing the specific cancer type, then one doesn't know which of the immune suppressive drugs have the potential to aggravate or not change, or may even be of benefit to that individual. One might consider using an antibody lowering product like an FcRn inhibitor. One could consider a complement inhibitor, which to my knowledge has no impact on cancer growth, cancer cell proliferation, etc. In all instances like this, I think it's critically important that the treating physicians talk to each other. The oncologist is in concert talking with the neurologist talking with the oncologist, and hashing this out. And this is what we do with our patients. I've just had a patient who had COVID with a persistent cough after recovering from COVID, a smoker, and asked her to get a chest x-ray because I was concerned about a pneumonia and has a mass in her lung and has cancer. And we've had to adjust her treatment paradigms and what we're allowed to use. In some instances, we have to weigh the balances. Does the therapy of one have the potential to impact the other? Knowing that up front, you may say, okay, go ahead and use it, because if the MG, for instance, gets worse, I have these options in play that I can consider. And this is what we've done with this individual, because her only hope was to use one of the newer forms of therapy for cancer that has the potential to aggravate in a small number one's myasthenia. But it's this interaction, this discussion, and looking at the potential options and pre-planning for the worst case scenario, if you will, that then allows you to somewhat comfortably proceed to treatment.
Thank you, Dr. Howard, for this excellent response. I mean, I'll inject a little bit my own kind of challenges with patients in myasthenia gravis. A patient whose thymoma, unfortunately, is becoming metastatic, for example, and despite radiation, there's growth of mass lesions not amenable to chemotherapy. Considering those novel therapies that can be life-saving is one of the considerations, but all the risks associated with them. In those scenarios, would you lean towards one or more of the novel mechanisms to try to optimize the patient and tightly control their disease beforehand, or would you be more reactive? In addition, of course, on top of that, a very detailed discussion with the patient, with the oncologist, a very close follow-up in that case scenario, you know.
Yeah, I think either of the two novel therapies we spoke about have the potential to be used here. Now, is one better than the other in this particular patient? It remains to be determined, but one could consider using either. And then again, taking into the account what are the other comorbidities, what is the financial wherewithal, et cetera, what are the logistical implications in initiating such a therapy? Do they have the strength, the stamina, the resources to come in for an infusion every 2 weeks or every 8 weeks or 4 weeks and then see what happens? All of these things have to go into the formula. And with the patient's input, of course, this is a two-way discourse, and the patient always has to be a part of the ultimate determination of what's done. But that would be my approach.
Thank you. The next question from the audience here is about antibodies and myasthenia gravis. Are there still antibodies that are associated with MG that are yet to be discovered? Are we getting closer to the discovery of novel antibodies?
That's a great question. And the protein structure at the neuromuscular junction is so complex. I'm sure there are other antibodies there to be discovered that may play a role in the pathology of myasthenia. However, I think they are going to be of very low, ultra low frequency. 85% of our patients have antibodies to AChR. Now we're talking about a little less than 10% have antibodies to MuSK, and less than 1% have antibodies to LRP4. And as we further identify new ones, those percentages will change a little bit. But I think we have found the majority, but I would not be surprised if we found 1, 2, maybe even 3 others, very small roles that can play some significant portion of their disease.
Yes, definitely. And these proportions are quite high in the generalized MG population, in contradistinction with the ocular ones, where we have more challenges with the antibodies. Thank you, Dr. Howard. The next question is about prevalence of MG, or the populations most affected by MG by gender and age. And it comes down to, is it more commonly diagnosed in women of childbearing age and men over age 60?
So I think that's changing over the decades. In the US, we have somewhere between 10 and 14 per 100,000. In the Toronto, Canada region, it's up to 41 per 100,000. And these are in part due to surveillance techniques. They have a very centralized database to extract information about clinical diagnoses, medications that might be linked, such as pyridostigmine, to use, and then can get numbers of patients. We don't have that in the US. We're too fractionated. If you read the textbooks, it says younger women and older men. Our data suggests that over the last several decades, from the 60s, when I started collecting this during my training, and now I'm seeing older men with the diagnosis. Does that mean the disease has changed, or men were macho, if you will, back then and didn't seek medical attention, and now we're starting to uncover it through social media, through changes in attitude, etc.? I don't know. There is some literature to suggest that autoimmune disease is increasing in frequency, not specifically MG, but autoimmune disease in general. And so it's quite possible that there is a shift, and that now it's more likely to be an older man who will develop the disease than a younger woman. In China, for instance, it's much more prevalent here in the US. And very interestingly, many of them have juvenile myasthenia. The largest prevalence of juvenile MG is in China. And so is there some genetic link in that population that is affecting their numbers? And we don't know that for yet. There is a large number of centers here in the US in concert with the NIH that looked at the genetics of myasthenia a few years ago, and we have not found a specific gene that causes myasthenia. We have found 4 genes that get upregulated, turned on, if you will, that relate to autoimmune disease. And so it's possible that with that genetic background, one is more likely to develop an autoimmune disease, but we still don't know whether you get MG, whether you get thyroid disease or lupus or rheumatoid arthritis. And, you know, I jokingly say it could be the phase of the moon at what time you develop symptoms that determines what you get. But that's an area that we don't understand, and we need further work. But no question, we have genetic prevalence to autoimmunity or autoimmune disease, but we don't know which one we get. And I think overall, we're seeing more men than women acquire the disease.
Thank you, Dr. Howard. The next question has to do with MuSK-MG. If a MuSK-MG patient were administered efgartigimod, could they cross over to a CAR-T-based therapy? That's the antibody-based therapy with CAR-T, if CAR-T proves to be helpful.
Oh, yes, I would think definitely. Now, would they be eligible to participate in the trial? That depends on how the trial protocol is written and what the inclusion/exclusion criteria are. I can tell you from my personal experience, I had a patient who was receiving a complement inhibitor who failed, and knowing the half-life of the drug and the duration of action of the drug, we went 5 times that length, believing that she was now devoid of any effects of that compound, and we enrolled her in our CAR-T program. And so for that particular protocol, it was acceptable. But it depends on the individual protocol of what inclusion/exclusion criteria would be. Efgartigimod, from what we know, has a limited activity, a limited life, if you will. And so one like plasma exchange, after a period of time, we would not anticipate effects of that drug. Now, some with FcRn, there may be some hints that there's other mechanisms that is yet to be fully proven. But at the moment, I would think that if one were on an FcRn inhibitor and then wanted to enroll in a CAR-T program, if it were allowed by protocol, after a certain period of time, they would so.
Thank you, Dr. Howard, for clarifying. This is very study specific. Each study will be designed differently and will lead to different outcomes regarding candidacy in research studies. Sometimes it takes adjusting the medication therapy to make sure someone would qualify, especially if they're very motivated to get into a certain study. The next question has to do with dalfampicin. This is the MS drug that's used for spasticity, I believe. So someone has used it as an add-on therapy, is having good results. And the question is, what other off-label but approved therapies are showing good promise, approved for other indications, I'm presuming, that's implicit by the question.
Well, I'm not specifically aware or knowledgeable of this drug, but everything in our toolbox, from cyclosporin to tacrolimus to cyclophosphamide to prednisone to methotrexate to mycophenolate mofetil azathioprine, are not approved for MG. There are FDA-approved drugs here in the US, but for other indications, and we use them in an off-label fashion. And we have success with them. That's been our mainstay since I was in training in the 1970s. And there are several others that people have used that have been approved for other indications in small numbers or on an individual basis with therapeutic benefit. The immune system is so vast, so complicated, intricate, that there are multiple drugs that could be used and would derive benefit from them. The keys are how long does it take, and what's the side effect profile? And these are the issues that, until we have large-scale data, we can't be sure of, for instance. I always worry about what are the off-target effects of any new drug that is being thought about to use MG. Do we target C5, or do we go upstream and target something higher like C3? And we have an active trial with a anti-complement drug targeting C3. It's a pill taken twice a day. And theoretically, it should work. So a trial has been created, and we're studying that now. But as we go upstream, then we have to be concerned, what are the off-target effects, side effects that we might encounter? We may encounter none, or there may be numbers of them, which would then dictate, how useful will it be? And so, again, this is the role of a clinical trial. We take someone who has a concept, an interest, we convince somebody, either an investigator, like Mazen and his team with Mamantha Pasnur did with methotrexate, or we go to pharma like we did with eculizumab and then ravulizumab and efgartigimod, and then put it into clinical trial to see the results. Clinical trials are artificial. we have to define very discrete inclusion-exclusion criteria. We have to sort of pick our patients. And the reason is that typically you get one shot to prove something. And if you fail, it goes on the back burner and we don't consider it further. And these trials are hugely expensive. Some of them approach $1 billion to execute in MG. And so it's not money that is free flowing and allows a company, for instance, to say, well, if we tried it in this group, can we go try it in this next group? And so we have to plan these. We have to stack the odds that we're going to see an effect. And then the FDA approves it, and they approve it for the generalized disease, much like Regain eculizumab was. But then the payer says, no, you can only use it in this population, which I think is wrong. And that's the card we've been dealt. I sometimes tell folks that physicians don't practice medicine. Insurance companies practice medicine. I may get in trouble for saying this, but it's a problem. And we have to figure out ways to ameliorate this effect. If it's approved for a wide population of patients, you need to be able to use it as such.
All right. Well, thank you. And then that kind of addresses the next question, but I should mention the comment in that question about your presentation. It says, wonderful update. Thank you, Dr. Howard. Of course, thank you. It's a great update.
I appreciate the opportunity.
As studies are being designed, I have a question, and we'll go back to audience questions. One of the things that Carolina Barnett has been championing, looking at the impact of non-muscular symptoms in MG, of impact of therapies on those measures like fatigue, for example. And then also, one of the things you mentioned is the pool of MG patients that participate in research studies ever shrinking because some participate and you're looking for the next patient. So there's an opportunity in those watershed areas to reach out to them. What's the potential of including telehealth, telemedicine as part of participation in research studies? Because I know you're on the forefront of designing studies, so probably the audience would be interested in knowing what kind of efforts are being invested in that direction.
Yeah, so study recruitment is a real issue. And at the last MGFA meeting, we presented data that shows the bulk of the patients that are recruited for clinical trials come out of California, Texas, the Northeast, and North Carolina. And the rest of the country is, it's small pickings. And so we're going to have sponsors fly patients in, or we have to go to the patient to do the evaluations. And, you know, neither is practical, it's expensive. And so telehealth then becomes a potential. Amanda Guidon at Mass General has developed a telehealth, a fairly objective clinical examination that to try and standardize things. That was published several months ago, and this came out because of COVID and the pandemic and the lockdown that we all experienced. The problem is trying to convince the FDA to use this as a primary outcome measure. The MS folks have experience in this, and it took almost a decade to change one of the primary outcome measures from what they've been using to one that they currently use. And so it's a very laborious process. The other thing that we're looking at, and we're initiating a trial shortly using a cell phone app that will look at things like ptosis. It will detect changes in speech. It will look at how long one can hold their arms in an outstretched position before they drop a few degrees. So we can use this as a timed task, if you will. Other apps that are under development look at activity rates in terms of standing, walking, sitting, sleeping. Now, all of this is predicated on one's willingness to wear a device on a chronic basis and literally not take it off if we were to use it in a trial. And so that's the potential downside of it. But it's an area, it's a hot topic. I listened to a lecture a year ago of an individual, of a scientist developing an artificial intelligence program that can detect simply by facial recognition whether you have atrial fibrillation or not. Don't ask me how he does it. I have no clue. But they had high hit rates. And so computer technology clearly is the wave of the future. We're still in its infancy, but it's forthcoming. And so if you have the capabilities of participating in such trials, and there are a couple of them going on, by all means do so. Several of the companies have apps that look at quality of life measures, ADL, etc. Argenx has one, UCB has one, that are providing longitudinal data on verbal outcome measures from a patient perspective that will be helpful using real world data over time to look at trends. And do we see signals? Can we then pull out that kind of information and use it from a regulatory perspective? Only time is going to tell.
Thank you, Dr. Howard. A question about pediatric myasthenia gravis. A couple of questions, actually. One of them seeks advice for a 12-year-old who has MG, the concern about deterioration of the nerve receptors. We're talking about those acetylcholine receptors kind of being degraded. And then the question is about complement can regenerate those. The patient currently is on Mestinon and IVIG. If the receptors go away, then how is this impacting the treatment?
Well, I didn't say that complement regenerates. That's a hypothesis. It's something we're actively the community is actively looking at, and to see if that is indeed the case based on the data that we have. Pediatric MG is difficult to treat because we expose our children to drugs that we really don't want to. We don't like to use corticosteroids because they may alter growth. We don't like our long-term immune suppressants because we really don't know what happens in terms of fertility, childbearing over decades of time. But in some instances, we must in order to keep the child reasonably healthy, out of the hospital, alive for that matter. But there are these pediatric clinical trials coming. The eculizumab trial is finished. There will be a trial with ravulizumab. There is an ongoing trial with efgartigimod, Vyvgart. If you live near North Carolina, we'd be happy to see if you qualify. We're searching for patients. And as drugs come out and become approved, the European Medicines Agency, the Europe equivalent of our FDA, mandates that a small number open label pediatric trial be performed. And so if you have the opportunity for your child, this is a potential way to have access to one of our newer therapies that we've already shown works in adults. And now we're trying to show that we see similar responses in children. And they're all open label to date, may not be in the future, but at the moment, these are all open label studies, so everyone gets the drug. And so that's something to keep in mind and consider.
Thank you. There's a question about Vyvgart use and in context of an upper respiratory infection. Someone's due for their infusion. They have a cold, stuffy nose, no fever, no excess phlegm, just a slight non-productive cough. Would you delay the infusion or would you proceed as planned? Or is that a discussion with the primary care provider?
Yeah, that's clearly a discussion with the primary care provider or the neurologist. Having never seen any of the records or seeing the patient, listen to the lungs, etc., then I can't give advice in this regard, but have a discussion with your treating physician and they can determine when it is safe, if not now or when, to receive efgartigimod.
And you did touch on this next question, Dr. Howard, a little bit, but particular to musk, there's a little bit of disappointment that there's no labeling for musk MG in the way of novel therapies. What do you see in the pipeline that's likely coming to benefit musk-positive patients?
So yes, it was disappointing that the FDA did not give us approval for non-AChR antibody negative patients. Japan has, and they're using it. I will tell you that the company is very active in trying to assemble data that we can take back to the FDA and try and get a labeling change. We will use the Japanese experience in these AChR seronegative patients to support this. We are going back and looking at longitudinal information of seronegative patients. Will we do a study in seronegative all over again? I don't know. There's a cost issue, et cetera. And I can't speak for the company per se, but they've not abandoned nor have they forgotten about the non-AChR positive patient, the seronegative patient. So there's active work going on there for efgartigimod. There are other clinical trials that are recruiting MuSK patients. One is the Cartesian Descart-8 CAR-T program. The other is the Cabaletta Bio CAR-T program. I think inolizumab is recruiting MuSK patients. There are trials that are being developed by at least 3 other pharmaceutical companies that will include MuSK patients. So stay tuned. They're not available. They're not out in active recruitment at this time, but will be so hopefully in the next several months.
Excellent, thank you. There's a question here. That's question number 16. Is it a fair assumption that all these new medicines will remain very expensive?
So that's the elephant in the room, cost. And, there's no way to predict. I have no input on cost. But think about it this way. Eculizumab, which at one time was the most expensive drug we use, it's now been supplanted by others that we use in the neuromuscular space for like spinal muscular atrophy, et cetera. They're generation 1. And we now have generation 2 drugs coming that will be a little bit cheaper. And then when we get to generation 4 or 5, the hope is that they will be cheaper yet. That's obviously not a guarantee. But all of the development up front is costly. And I'm not to say the price that they're using is appropriate, because I think all of these drugs are quite expensive as well. But one of the issues that is never taken into account is what is the true cost to treat myasthenia? And I was hoping that I gave you a flavor of this. And so if I put you on steroids, for instance, which is a relatively cheap drug to use, but if I give you diabetes, now what is the lifetime cost to treat your diabetes? Or if I give you osteoporosis, what's the lifetime cost to treat your bone disease? What's the cost if you take 30% of your time away from work because you're weak or having an exacerbation? And if I use a drug and can keep you employed, and I showed the employment numbers, 58% in Thailand, in Australia nearly, and 30% in Japan and elsewhere. That's unemployed. But if I can keep you employed, what's that cost worth? And then the biggest one is, what's the cost of quality of life to allow you to interact with family, to engage back in your sports, et cetera. And that cost is not zero. But none of this gets factored in to the evaluations that are done by groups like ICER and Hancock, when they say drug X has to be discounted some $150,000 to be applicable. They're only looking at cost of medication, cost of a hospitalization, but not any of this other stuff. And so I'm not defending pharma at all, but I'm trying to bring some reality to the program here as to, what is the true cost? And we don't have a good handle on that in the US. Our healthcare system is so fractionated that we cannot get good health economic data. And we desperately need that to look at the total picture. As I said, one of these trials in MG cost nearly $1 billion to run. And that's hugely expensive. There are a lot of moving parts to running a clinical trial. It's not simply the sponsor and the investigators out in the field. There are middle managers, so to speak, that do the conduct of the trial. There are separate laboratories that do the analyses. There are freight companies, airline companies that are flying specimens back and forth. And because I'm drawing blood from you for a particular study at day X, they aren't done locally in some instances. They're shipped to Europe. And specimens from Japan are shipped to the US or to Europe. And so there's costs there. So you're quite right. Cost is high, and we can't afford this as an ongoing basis. But I can't predict what these medicines will ultimately cost. My hope is that new generation therapeutics within a disease class will be much cheaper than the first one that we've brought to market.
Thank you for this eloquent response, Dr. Howard. Someone is interested in seronegative MG patients, and their question about drugs other than pridostigmine that are available for seronegative patients. Would you like to clarify the approach to seronegative patients?
Yeah, so seronegative patients is one of the true unmet needs in MG. And while the seronegative patient represents a small number of the total MG population, it's the largest patient group that I see when I was in active practice, 5 days a week, so to speak. I think it's a difficult diagnosis to make. It's a diagnosis that is often misdiagnosed. I think sometimes even neurologists make a mistake, myself included. I had a patient that I inherited that I treated with plasma exchange and mycophenolate and rituximab for several years. And she clearly had some improvement, not tremendous amounts, but some. And so we kept on. And when genetic testing came available, we did it, and she has a congenital myasthenic syndrome. And so we changed course completely, and it's situations like that. But if one is truly seronegative, we use Mestinon. But in many instances, we're able to obtain approval to use drugs like mycophenolate or azathioprine or IVIG and even plasma exchange. At the moment, they're not eligible for our complement inhibitors, and we don't have approval in the States for FcRn use, but we're hoping to change that. I think it would be interesting to know whether complement inhibition works in seronegative, and there are scattered case reports where it has shown benefit. And there are rare payers, insurance companies, that have allowed its use in that particular patient, but it's not widespread. I think we have to do a better job at educating these payers for this. I think we have to try and get all future trials going forward to have a small number of patients who are seronegative and not use them in the primary analysis, because we have to make sure we can see a change, but to be able to see a signal that then may allow us to go and do another trial in this AChR negative population. And I think we have to insist that this be used. Now, will all sponsors do that? No, because of budgets, because of logistics, etc. Recruiting seronegative populations into clinical trials is hugely difficult, in part because there's so few and they're scattered all over the country and we don't have ready access for them. So again, my plea is that if you are seronegative and you have a trial within your locale that is running a study that includes them, step up and participate. Or if you live away, contact the sponsor. Clinicaltrials.gov is an excellent website. You can search for trials in MG. You can see what the inclusion criteria are, and they have a contact number, usually at the sponsor site. And so if you see they are recruiting AChR negative patients, contact them. Tell them you live up in, who knows where, and you're eager to participate, and they'll try and accommodate you into a trial site that's near you.
Thank you, Dr. Howard. Maybe we'll take 2 rapid-fire questions. One of them is about aging, distinguishing aging symptoms from those of MG. That's one aspect. The other question has to do with what about the placebo group? Did they get the same treatments in the way of prednisone or other in studies here? This is specific to Vyvgart study.
Yeah, so the latter is the easiest to answer in that the placebo population was very evenly matched to the active drug arm. And then, of course, if they completed the study, they rolled over into the open label and got active drug. So yes, they were matched for standard of care. The other is a very interesting question, and what is the effect of aging? And as we age, we lose muscle mass. We call it sarcopenia. Our outcome measures are designed to try and account to some degree for this. It's not perfect, but we're trying to do our best. But no question that the individual who has lots of aging, atrophy, if you will, as well as all the other comorbidities of aging, has an impact on our ability to get good data in some instances.
Thank you very much, Dr. Howard, for this excellent presentation, excellent discussion. It's been really a tremendous pleasure to moderate with the expert, the ultimate expert in the field. And thanks for the participants for these wonderful questions.
Yes, I echo that. Thank you for your superb questions, and I hope we've been able to answer them for you. Thanks for the opportunity to be a part of this. Allison, thank you. Mazen, thank you.
At this time, we'll turn it over to Michael and Allison. I think they have some presentations for us.
Thank you so much. You are both experts in the field, Dr. Dimachkie and Dr. Howard. Thank you for spending your morning with us. We have a few more messages from our partners that we'd like you to tune into. I am going to invite you to hear from Immunovant and JB Smalley.
Good morning. My name is JB Smalley, and I'm the head of patient advocacy at Immunovant. And on behalf of Immunovant and all of my colleagues, it is a pleasure to be here today with you for the MGA's 62nd annual meeting and educational seminar. So some of you may not be familiar with Immunovant, and so I wanted to make sure and introduce you to Immunovant and the work that we're doing in myasthenia gravis. So our vision at Immunovant is normal lives for people with autoimmune diseases. And we are boldly developing anti-FcRn technologies for a range of debilitating autoimmune diseases with significant unmet need. We truly believe that to have a patient-focused drug development approach is vital to meet the complex and variable needs of people with autoimmune diseases such as myasthenia gravis. We have 3 core values that shape our work at Immunovant. First, we love trailblazing. We're constantly rethinking possibilities and challenge each other to create breakthroughs. We also take a bolder, faster approach. We take action and we move quickly and with confidence to ensure that people living with autoimmune diseases are empowered. And finally, we embrace all voices. We listen to and learn from the patients we serve and our colleagues because we are better together. If you'd like to learn a little bit more about the work we're doing in myasthenia gravis, you can learn about our phase 3 clinical trial of batoclimab in MG, the FLEX study, by logging on to our website that's listed here. Most importantly, and on behalf of Immunovant, I'd like to thank the MGA and the members of this incredible community for their partnership. You have let us into your lives. You have shared your stories with us. You have advised us on what it means to have truly patient-focused innovation in MG. And you've partnered with us on a multitude of awareness initiatives to help raise awareness of myasthenia gravis. We are so grateful for this partnership. And we hope you have a wonderful meeting today. And thank you for your time and attention.
Hi, my name is Dr. Omar Sino, U.S. Medical Strategy Lead for Rare Disease at UCB. So people with rare disease often feel forgotten, unheard, or misunderstood. The challenges they face can be magnified by isolation, and they may wonder if anybody else cares about what they need. They worry their disease affects too few people for anyone to care. And we know the greatest needs can't always be measured in numbers. So at UCB, we don't just see patients or population sizes, we see people in need. Through decades of serving the neurology and immunology communities, we have improved lives with impactful medicines and by enhancing the social and emotional well-being of patients. We are driven by science to translate patient insights into unique, disease-modifying solutions that provide value and transform the patient experience. Optimizing patient experiences at every touch point with our company and our medicines is integral to everything we do. Our decisions matter because moments matter. And we know our decisions impact the big and little moments that are important to patients. In speaking with patients and caregivers, they tell us that they want to share those everyday moments they sometimes miss because of their disease. From cooking a meal with their family, to playing with their kids, to having a conversation. We are inspired by these patients and the ways they overcome those difficult moments in their disease journey. They demonstrate empowerment and resilience, and it is essential to partner with MG advocacy communities to build momentum for identifying unmet needs and improving health and care for people living with myasthenia gravis. Collaboration is the cornerstone of any effort to improve life with MG, and this is based on a shared vision and purpose. The time is now to tackle the issues that burden those living with MG. Thank you so much.
Thank you so much, JB and Dr. Sano. We appreciate hearing messages from all of our partners and the work that's being done in the myasthenia gravis space. Next up, we're going to hear from our friends at Janssen.
Hi, my name is Sindhu Ramchandran. I'm a neurologist and neuromuscular specialist and the clinical development lead for myasthenia gravis at the Janssen Pharmaceutical Companies of Johnson & Johnson. At Janssen, we are delighted to support the 62nd MGA annual meeting and educational seminar. We feel privileged to support you today and every day in your myasthenia journey. We know that living with MG and autoantibody disease can be challenging and that there is still a tremendous unmet need for more treatment options. We are committed to working hard in partnership with the MG community. We have engaged with people living with MG, and we incorporated your insights and real-world perspectives and what is important to each of you in the design of our ongoing clinical trials for seropositive and seronegative MG in adults and for seropositive MG in children. The goal of our research is to help all patients with MG. We want to continue connecting with you, and we encourage you to visit imaginemymg.com, our online community with Janssen, which has resources and opportunities to interact. Today's event is critical to sharing information about emerging treatment options and research in MG. Janssen is proud to partner with MGA and each of you as we work to reach our common goals. Thank you.
Thank you so much for joining us this morning. Next, we're going to hear from ARJ Infusion Services.
Prompt Care is a home infusion specialty pharmacy Home infusion is an opportunity for patients to receive their medication in literally the comfort of their home versus a hospital setting or a suite setting. And the nurse will actually go into that home, teach the patients how to administer that medication and or, depending on the type of therapy, administer the medication and monitor the patient.
We're an infusion pharmacy, so we have to supply drugs and IV drugs for our patients that are in the home care setting. The drugs that we make are not oral drugs, they're infusion drugs, IV drugs, and they're high tech, and they have to be specifically mixed in a clean room and packaged.
Patients that receive these medications are clinically either nutritionally imbalanced or they need antibiotics. It's more of a high-tech therapy that you would receive in a hospital, but in a home setting. We need nursing, we need nutritionists, we need the doctors. We review, monitor, and dispense depending on that prescription and with the collaboration of the physician.
With COVID and all of the problems that it has created, having prompt care is really helpful because a lot of patients don't want to come to a hospital and they'd rather be treated at home. This is a wonderful avenue for me to have as an additional way of treating my patients.
So at Prompt Care, our philosophy is doing primary care nursing. The patient will get assigned a nurse who will follow that patient throughout their course of therapy. Home infusion has really opened the door to customized care for an individual patient. I feel as though I give more to myself to a patient. It's nice knowing that I'm not being split up during the shift of a hospital. The nursing care that we give at Prompt Care enables our patients a quicker recovery in their own home setting.
Prompt Care has a lot of moving parts to administer this medication and get this medication into the home. We have about 100 field nurses who actually give medication and take care of our patients, communicate back to the physicians.
Since COVID, there was a lot of fear and confusion with the patients. We had patients that were going into the infusion suites at some of the main hospitals in New York, bringing their children for their infusions monthly. And they said, I'm nervous, how can I get this at home? So we innovated a process to be able to get their drug at home so they could do it safely and not worry about travel or exposure by going into these hospitals during this pandemic. We've been able to help so many patients now, and keeping them out of the hospital and safely at home keeps the hospital beds open to treat patients.
They tell our pharmacists, our nursing staff, how we're helping them to get through their daily lives.
Prompt Care got involved, and I'm telling you, I have a life again. They're a great team. They've helped me.
Before he started his IVIG treatments, he could not function as a normal 7-8-year-old kid. And now it's night and day. We know that when he has his infusions, he can go back to a normal life and be a kid again.
Prompt Care has been the only home care infusion company that we would use.
Thanks to Prompt Care, my burden has been eased. I agreed to do this because I want to support a company for all they've done for me to make my life easier through the pandemic and through this disease.
They sent us a wonderful angel named Bree, who is supportive of our whole family. She's been training me to do the infusions, and she's there 24/7 without a doubt. She answers every call I make.
You walk away and your heart is full because you know that you're helping somebody with a disease that they have.
Because we've been around 30 years, we have great relationships with the marketplace, with physicians, with insurance companies, with pharmaceutical companies. Because of our relationships, we're really able to handhold all of our referrals and our patients from, start to continuing on service and really kind of offer a care management program for the patients.
When I hand a case off to Prompt Care, I expect a quick response. They're going to solve the problem for me. This gives me peace of mind, and it gives my patients peace of mind.
We're a mission-focused organization. Everything at Prompt Care is patient first. Patients with these long-term chronically ill needs, many of whom are children, and the people who work at Prompt Care, the most caring and committed and compassionate people who are engaged in a calling that's greater than themselves.
Hello, everyone. My name is Amy Grover, and I am the Senior Director of Patient Advocacy at Catalyst Pharmaceuticals. I want to thank Allison for inviting me here today and give you a brief update. Catalyst Pharmaceuticals is a commercial stage biopharmaceutical company focused on in-licensing, developing, and commercializing novel medicines for patients living with rare diseases. With exceptional patient focus, Catalyst is committed to developing a robust pipeline of cutting-edge, best-in-class medicines for rare diseases. In my role at Catalyst, I continue to work with the Lambert-Eaton myasthenic syndrome community, offering tools, resources, and most importantly, much needed awareness for this ultra-rare disease. For those of you who don't know, Lambert-Eaton myasthenic syndrome, or LEMS, affects roughly 3,000 people in the United States and is closely related to myasthenia gravis. In fact, 30% of LEMS patients are misdiagnosed with MG first, making it a challenging diagnosis. Approximately 50% of patients diagnosed with LEMS are also diagnosed with an underlying cancer, which is usually small cell lung cancer. We are currently working with the lung cancer communities to raise awareness for LEMS. In 2022, we launched a new podcast on the LEMS Aware website. I invite you all to listen to this podcast as host Kevin speaks with patients and caregivers on the challenges living with a chronic disease like LEMS. You can find the podcast on the LEMS Aware website at www.lemsaware.com. Congratulations to the Myasthenia Gravis Association on another successful and informative event. We are honored to partner with you as we all raise awareness for neuromuscular diseases like MG and LEMS. I hope everyone enjoys the conference.
Thank you so much. You may have noticed that ARJ Infusion Services used the company name of Prompt Care in the video. Depending on where you live in the country, they have transitioned to Prompt Care. So we appreciate their continued partnership as their reintegration moves forward. Also, thank you, Amy, with Catalyst. We want to move into the last part of our meeting today, and that is our awards ceremony. We have 3 awards that we're giving today. Our first award is the Stackhouse Distinguished Service Award. I had the honor this year to meet our founders, the Reverend William and Joan Stackhouse. This award is our most prestigious award given to recognize outstanding leadership, dedication, commitment, and furthering the cause of supporting all those affected by myasthenia gravis. It is presented to highlight exemplary spirit of volunteerism and leadership like that of Joan Stackhouse and her husband. This year's winners are Sandy and Gary Gardner. The MGA is excited to present the award to the Gardners, and they will get the award at our upcoming holiday party. Their awareness of myasthenia gravis came when Sandy's late brother was diagnosed with MG. Years later, Sandy would become diagnosed. For over 10 years, Sandy has been the leader of our Kansas City Northland support group. Not to be stopped by a cancer diagnosis or a pandemic, Sandy has continued to find ways to connect with members of her group, whether it's roping Gary into giving boat rides on Weatherby Lake or hosting a table at our trivia night. Sandy and Gary have not only been hands-on supporters, but financial supporters of our organization. Congratulations and thank you. The next award we want to give today is our Volunteer of the Year award. Our Volunteer of the Year is somebody who dedicates time and resources to our organization. And this year's winner is a true picture of a Volunteer of the Year. The MGA is excited to present Keith Asali with the Volunteer of the Year award. Chances are, if you've been involved in our MGA Triple Crown Showdown or received a new patient packet in the mail, Keith has had his hands in it. Keith has been a volunteer for the MGA for over 8 years. He often ropes in his large family. You've probably seen them taking down tents and tables at our 5K run in the pouring rain and stuffing packets at our office. We are so thankful for Keith's longstanding support. Congratulations, Keith. The last award that we want to give today is an award that we rolled out a couple of years ago when our longtime volunteer, Diana Wilmoth, retired from her position as a volunteer in our St. Luke's clinic. This award is not given yearly. It's one that we give every couple of years when we feel somebody has made a lifetime difference to our organization through their leadership, their giving, their volunteerism, and spirit. They've helped others with myasthenia gravis to reach their full potential. This morning, the MGA is pleased to give the award to Dr. John Sand. I think this is the first annual meeting in probably close to 30 years when he hasn't been here. You can see why he is so deserving. Dr. Sand has worn many hats within our organization on the board of directors. He played a crucial role in establishing our clinic partnerships, maintaining them, and keeping the doors of our organization open. You'll likely recall him from his many years of moderating and being visible at the annual meeting. He's also remembered for roping his family into volunteer roles on committees and boards. Dr. Sand's vision to support patients has never wavered. While he has stepped down from our board of directors, he is so well deserving of this award and honor. Thank you, Dr. Sand, and congratulations. This concludes our 62nd annual meeting and educational seminar. Don't forget to check our calendar for updates and offerings to our support groups. Our virtual monthly meetup is next Monday, November 21st from 6:30 to 7:30 p.m. Central Time. We are going to learn more about plasmapheresis and the process that it does take in treating myasthenia gravis. Also, we have several in-person groups happening in different areas. Be sure to check your email. Again, our website, mgakc.org, Facebook events to see what's happening in your area. And thank you again in advance for completing our survey on today's program. Thank you very much.